Hello, my name is Vanessa Espinoza, and this Epidemiology course project is focused on neurological disorders in relation to herbicide exposure. The table of contents is just an overview of what is to be covered through this presentation. What will be covered is a definition on what is an herbicide, what are neurological disorders, an overview of descriptive studies on neurological diseases, etc. We will have a quick brain exercise quiz or game to test out your memory, as it ties in with the discussion on Alzheimer's. You will have a better understanding once we get to this part of the presentation. Then we will focus on the research study and design for the remainder of the presentation. What is an herbicide? Herbicide, or herb, is Latin for herba, which means a flowering plant whose stem above ground does not become wood-like, or an herb is a plant. Side is Latin for sida, or killer, act of killing, used in the formation of compound words. Those compound words are as follows: nematodicide, insecticide, larvicide, fungicide, and rodenticide. Herbicide is a chemical designed to control or to destroy plants, weeds, or grasses. The definition by the Environmental Protection Agency, EPA, depicts the meaning of herbicide and pesticide by defining herbicide as a chemical designed to control or destroy plants, weeds, or grasses, while a pesticide is a substance or mixture of substances intended to prevent infestation. What are neurological disorders? Neurological diseases are disorders of the brain, spinal cord, and nerves throughout the body. When something goes wrong with the part of the nervous system, you can have trouble in moving, speaking, and swallowing, breathing, and learning. Problems with memory, sense, or mood may occur. There are more than 600 neurological diseases. Some are as follows. Huntington disease and muscular dystrophy are caused by faulty genes. Spina bifida occurs when there is a problem with the way things develop. Studies have shown that spina bifida can also occur when a woman is exposed to herbicide and may produce this defect in her newborn. Generative diseases, when nerve cells are damaged or die, such as Parkinson's disease and Alzheimer's disease. Diseases of the blood vessels that supply the brain, such as a stroke. Injuries to the spinal cord and brain. Seizure disorders, such as epilepsy. Cancer, such as brain tumors. Studies have also shown that herbicide exposure may cause cancer, as we will touch base on later in the presentation. And finally, infections such as meningitis, which is an inflammation of the membranes that cover the brain and spinal cord. This is a descriptive study on neurological disorders. This gives us a general overview on the few but most prominent of all neurological disorders. One that is very common is Alzheimer's. Here the person are people ages 18 and up, and the location is in the United States. Neurological disorders affect people in all countries irrespective of age, sex, education, or income. You may wonder how this relates to herbicide exposure. Well, analytical studies have shown that herbicide exposure may have long-term damaging effects on the nervous system that contribute to the development of Alzheimer's or other dementias. Let's review a background of Alzheimer's. Alzheimer's is the most common type of dementia. Most Americans are 65 and older, which is considered what is called a late onset. 1 in 8 million Americans have Alzheimer's, which accounts for 13%. An estimated 5.4 million Americans of all ages have Alzheimer's disease in 2011. 5.2 million Americans 65 and older, and 200,000 individuals under age 65 have AD. This is considered what is called a younger onset. Of the 5.2 million people age 65 with AD in the U.S., 3.4 million are women and 1.8 million are men. 4% under 65 have AD, 6% are between the ages 65 through 74, 45% are between the ages of 78 through 84, and 45% are 85 years old and beyond. More women than men have Alzheimer's. Almost 2/3 are women. This demonstrates a descriptive analysis of the proportion of people in the U.S. who are diagnosed with AD. People are white, African American, and Hispanic people. Location is taking place in Washington Heights in Wood Columbia Aging Project, and time is in 2006. Older African Americans and Hispanics are more likely than older white Americans to have AD. Since we have discussed a brief overview of Alzheimer's, let's test how good your memory is. Memory quizzes are healthy to challenge your brain to different stimuli and puts your brain to be focused. Material needed are a pen or pencil and paper and a stopwatch. I will present you with a table of 25 different objects. Look at them for 30 seconds, and after the 30 seconds the picture will disappear and you are to write down the objects you can remember. Here are the 25 objects. How did you do? How many did you remember? When I completed this brain exercise, I only remembered up to the coffee picture, and that's probably because I enjoy a good cup of coffee. Anyway, well done. Thanks for playing. Maybe next time you can remember more pictures. Continuing on, we are to focus on descriptive and analytical studies that are currently published and have already focused on neurological disorders, which is our health issue for this presentation. In a descriptive study, the researcher focuses on people, place, and time. An analytical study measures the how and the why. Comparison of dementia with Lewy bodies to Alzheimer's disease and Parkinson's disease with dementia is a descriptive study because it is measuring the distribution of Lewy bodies to Alzheimer's and Parkinson's disease. Pesticide-related illness and injury acute is analytical study because it is analyzing acute adverse health effects resulting from exposure to a pesticide product. This study also utilizes evidence supporting a causal relationship between pesticide exposure and health effects. Parkinson's disease and exposure to agriculture work and pesticide chemicals discusses the direct effect of exposure and outcome. The example shown here is how in this study agriculture workers are exposed to pesticide chemicals that is directly associated with the development of Parkinson's disease, and cancer mortality in workers exposed to 2,3,7,8 tetrachlorobenzo-p-dioxin, or TDD, also demonstrates a direct relationship to exposure and outcome, another analytical study. So for this study, the research question is, does herbicide exposure cause neurological diseases amongst at-risk agriculture workers in the lower Rio Grande Valley? This research will cover 4 counties, which are Star, Hidalgo, Willacy, and Cameron counties. So why an herbicide exposure study? Studies suggest that this chemical may be related to a number of cancers and other health effects in humans. Also to develop preventative care measures to better protect agriculture workers from herbicide exposure, to lower the costs driven by herbicide exposure that cause neurological disease and other health effects, to strategize alternatives of the product herbicides that will place humans at low risk of developing neurological disorders, and to raise awareness emphasizing the effects of herbicide exposure worldwide. Here is some information about the Rio Grande Valley. The valley is home to just over 1 million people. There are nearly 90% of the population is Hispanic. The RGV, also known as the Valley, is a permanent home to one of the largest concentration of farm workers in the U.S., and roughly 1/3 of the valley's population of over 1 million depend on employment in agriculture. The study design is a cohort study design. In a cohort study design, the investigator selects a group of exposed individuals and a group of non-exposed individuals and follows up both groups to compare the incidence of disease. You can see this by the examples shown on this slide. This study will serve as research on the impact herbicides has on the human brain and how much exposure from herbicides can cause a neurological disease primarily in the central nervous system. The variable for this study are the exposure, herbicides, and the outcome, neurological diseases. The independent variable, herbicide, will be measured biweekly with urine samples to identify pesticide metabolites during the time pesticide spraying is being conducted. The dependent variable, neurological disorder, will be measured by tests that demonstrate cognitive and neuropsychiatric effects, motor dysfunction, other CNS disorders, and peripheral neuropathy defects. Participants will be selected from the 4 counties. Once again, those are Hidalgo, Cameron, Willacy, and Starr County. The number in parentheses are the number of people living in those counties according to the U.S. Census. Participants will be randomly selected from the Texas Department of Housing and Community Affairs, TDHCA, list of housing agriculture workers. The sample size will be 4,000. What is a confounder? Whether factor A is the cause of disease B, or if there is a third factor, factor X, that is both a risk factor for the disease and is associated with the exposure. In this case, the confounder will be lifestyle, sex, and age. Lifestyle can have an effect or act as a third variable to this study because the life of an agriculture worker is to be around herbicide exposure. In some circumstances, the life of an agriculture worker, there is not an option to not be exposed to herbicides. Sex is also a confounder because when it comes to a disease such as Alzheimer's disease, as people get older, more women than men are diagnosed with Alzheimer's. So in this case, age and being male or female can contribute to the outcome. For this study, we will use the 2x2 table identifying exposed, unexposed to those who have either the disease or do not have the disease. Relative risk formula will be used to calculate the cumulative incidence exposed to the cumulative incidence unexposed. This is used to find the increase or decrease in risk caused by the exposure under study. The descriptive analysis will consist of the Hispanic agriculture workers in the Rio Grande Valley during the years 2012 through 2015. The analytical analysis of the study will be a prospective approach where the exposure is assessed at the beginning of the study and the outcome is followed into the future. A cohort study offers some biases. Some that might reflect off of this study are information bias. That is where information is obtained from past records. We don't know the history of these participants if they have already had a developing neurological disorder. Biases from non-response and losses to follow-up. This is due to when some participants throughout the course of the study decide to no longer participate or are non-responsive. This can complicate the interpretation of the study findings. The key strengths are that the study will outline 4 of the major counties in South Texas. Its main focus is on Hispanics. Study allows a direct estimate of risk and rate of disease occurrence over time. This study will allow the investigator to assess multiple outcomes of a single exposure. Avoid recall bias, which means as the exposure is determined before the outcome, one's disease won't affect how accurately one recalls exposure levels. Key weaknesses of the study is time-consuming. It may be costly and expensive. Some limitations is that maybe TDHCA may not disclose of all participants. Differences in quality of measurement may not equal to or may result in a distorted outcome. Here are some recommendations and tips on how to reduce herbicide exposure, whether it be around your home, yard, or for agricultural workers. It is a good idea to take preventative measures to reduce this exposure. Try to get rid of pests without chemicals. Diatomaceous earth is an odorless, non-toxic white powder that can be used on all types of insects and is much less harmful than herbicide chemicals. More information can be found with this provided link. If using pesticides, follow the directions. Store and dispose pesticides properly. Wear protective clothing, which includes long-sleeve shirt and pants, gloves and shoes, goggles, and a dust mist filter mask. Store protective clothing away from living spaces. Wash exposed skin, rinse gloves, shoes or boots. Change clothes after applying pesticides. And wash clothes soiled with pesticides separately from other laundry and hot water and detergent. The following are the references that were used throughout this presentation. This concludes this project. I hope you enjoyed it. Thank you very much.